protein function. As you know, Jonas will record, and today I will talk about the introduction, so terms. Many of the slides come from Jan Fran about function prediction, so the first 2 days. Others from Marco Punta. Jan Fran is now a professor at Berlin University. Marco Punta is at PFAM at the moment, the EBI. And others come from Karl Stroschinski. Karl Stroschinski, that is not true anymore. He's now a group leader in the New York Genome Institute. What do we know about molecular biology? So again, what is common to it, the genes, is one way of defining it. But ultimately what defines life is not the genes, it's a process. Biology says that you have replication, you have metabolism, you have cell division. All of these elements that adhere to these principles, they are life. Life is not defined as the one that carries DNA or is coming out of DNA. Biology defines life as a process. And that's a very important reality. It is not, in the olden days, Leonardo da Vinci had this idea, life is defined as some shape, some fitting into the harmony of the universe. The choir that you sing, it's one voice in this choir. And biology really defines life in its own category, if you want. Life begins from molecules. What is the precursor of life? How does life start? Suddenly there's life?
Maybe self-replicating molecules like RNA.
Like RNA?
I think in the beginning it was RNA.
Could it be anything else?
Yes.
What?
Maybe self-replicating proteins as well.
Oh, that's an interesting idea.
It's possible, as in prions or something. So it could have been something else.
That's an interesting idea. Yes, so RNA somehow starts it. Now, back to the idea of proteins. In the RNA world, do you need proteins? Yes. Exactly. To do the function. So those of you who have been with me in the lecture before, the theme, and it's going to come again here in the next slide, the dogma of the molecular biology. Here, at the beginning of it, RNA, in fact, uses proteins. So in that context, maybe prions could have done it, but actually turned out the other way around. In fact, in some sense, you may argue RNA is the master. Now, if that would be true, what would that imply? Could we test that?
Okay.
So the idea is RNA could not do its own enzymatic function, or not all of what it needed to do for self-replication and for whatever it did during its life cycle. For that, it took another chemical that was out there, proteins. If that is true, well, this is an assumption here, right? How would you sort of try to prove that assumption? Sitting today, I mean, not going back, yeah?
Well, we'd have to find an RNA molecule that can replicate itself.
So you could simply do it in a test tube. And that is true, you can, but that is true. But there's something else. Let's just assume you were a computational biologist. And let's just assume you don't have access to doing that experiment. Just in a computer. Any idea? What would it tell you? I don't know. What would it tell you about the proteins, the universe of proteins? Well, clearly, the RNA binding part would be something that you would reveal itself as a very ancient domain, right? Possibly even more ancient than protein binding DNA. And that is exactly the idea of André Lupas at the Max Planck Institute in Tübingen. And they, in fact, what they do is they go domain by domain and see the origin of that domain and categorize the protein structures according to their ancient domains that they argue were simply selected by binding RNA, and that what we see today in the universe of proteins, what we see today in terms of the amino acids, is clearly biased by that first reason for proteins to exist. Not to exist as the molecules of life, like a prion, like you say, but as the helper molecule for binding RNA and then later DNA. Yeah?
Can any of those structures form freely? Like
yes, that is one of those original domains here.
So if
it's essentially funny that you say that. It's a Greek, so-called Greek key motif. And it's a very needed minimal number of amino acids. You can essentially form it with 20 amino acids and create a unit that is stable, and you just put a little bit of an embellishment, a little bit extra, and then bind RNA, yes. And it's a theme that you see in many, 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 many proteins in many organisms. Okay? But the regular way in which we see it today in the central dogma is that DNA is the code, RNA is intermediate, and protein is the machinery of life. Any dent in that picture? Somebody else. Somebody who, ideally somebody who hasn't attended last. I may have made that point before.
I haven't attended last lectures.
You have not attended?
Yeah, one only, but I cannot have the full picture of the...
That's good, so answer that question. Thank you. I understand that this is not what you were saying hello for. What's the problem? So here's a picture that I give you.
Okay.
What's wrong in it? Dent from dentist, the tooth, the hole.
Is the other way around the direction of building blocks, like, or I don't know. Maybe.
Anybody? Yeah?
There are non-coding RNAs, and there also is reverse transcription, so that's the other way around.
That's the major, the non-coding RNAs, that's the major thing. So the part that this in fact is an intermediate step, let's begin with this. Ribosomes, all the proteins come from the ribosomes. Ribosomes are constructs of hundreds of proteins, plus 40 or 50 different non-coding RNAs. And they're absolutely essential for the functioning of the ribosome. We wouldn't have proteins, wouldn't it be for the ribosome, wouldn't it be for the non-coding RNA? There are short sRNAs and RNAis. Those are important short regulators. Stretches that in fact put upon the genome, decide whether or not a protein is turned on, and we clearly believe this is one of the most important mechanisms. Is that all? Are there many non-coding RNAs, excluding these short ones? Are there many longer ones? So I said in the ribosomes we have in the ballpark of 40. Is that all? Yeah?
308.
Yeah, but they are, again, that is not really an enzymatic, so it's not something in the sense of a functional element of a cell. In the ribosome, a functional element of the cell is something that has an enzymatic activity, right? So I exclude those because essentially they are more like tRNAs. They are more like things in between somehow that have an important function, and they absolutely need to be there. But what I really mean now is longer constructs that are there for a long time and act.
Ribosomes.
Yeah. They are part of that, yes, but are there many? How many? So here is Phantom 3, it's a long time ago, almost 10 years, 9 years ago, and it was one of those papers with 143 authors, and essentially it asked how many, if we looked at mouse constructs, and what they had at their hands those days were 160,000 mouse constructs, and they simply asked how many of those do we have an experimental indication are functional as non-coding RNAs. So in that set, and that was eaten up here by the style change of my slides. In that set, they found about 20,000, or no, 16,000 proteins, which we believe is very close to the 20,000 mouse has. How many long RNA, non-coding RNAs do you believe they found in there?
5 times as many.
So you believe that, let's just put this answer into perspective. You speak up and say long RNAs are 5 times more important than proteins. And you stick to that statement.
Yeah, why not?
But maybe not more.
Do you really believe this is, what?
You did not say more important, but just 5 times as many.
So the number is higher.
But that's a high number. Really believe that?
I wouldn't have said 5.
What would you have said?
How many did they have in...
So again, they had about 16,000 proteins, so they started with 160,000 cDNAs constructs. And they mapped them to 16,000 constructs that they believed are really protein coding, and N that they believe are non-coding. Long RNAs.
I would say the same. Also 16,000.
That's a remarkable answer, and it's a remarkable truth, yes. And that's exactly the number it was. Almost entirely the same. And we still, so this is 9 years ago. We now, for many more, know exactly what they do in terms of the enzymatic activity. But we still don't quite know how diverse these 16,000 long non-coding RNAs are. How functionally important the diversity is. So for protein families, we know for some protein families we need several representatives of those families, right? For those RNAs, we still don't know that because they're difficult to follow up on experimentally. So there's a universe of non-coding RNAs. We don't believe it's as big as 5 times the proteome, but it is considerable, and we're going to ignore that from now on. Let's get back to protein function. Protein function is on many levels, so you see different protein structures, many levels here. What is a protein? We had that, so whoever was here before knows some answers, but who wasn't here before? What is a protein for you?
To explain.
One way of saying it is a sequence, right? But really, what's your image of a protein? Definition of protein. Is a gene a protein?
Sometimes.
Can you specify what you mean?
Well,
how can a gene not be a protein?
Pardon?
How can a gene not be a protein?
It could code for an RNA.
Okay. Good point. So a gene can code for more than one protein. Or it can code for things other than a protein, a non-coding RNA. There's alternative splicing. A gene can code for more than one protein. Is that all? Are there proteins that are coded for by several genes?
Well, if you count multi, like protein complexes, yes.
So, what do you mean by complexes?
Something like a nuclear pore, or
yes. So we are looking at something that, like ribosome. In an extreme case, is that reasonable to call a ribosome one protein? Would you, so ribosome?
It is a functional
unit.
Yeah.
Exactly. Would you call it a protein? No, you would call it a complex, right? Have you guys ever heard of ATP synthase?
Yes.
That is a protein, right? And that is spliced together by genes from different chromosomes. It's a big thing, but it's nowhere near a big complex. It is something that when you look at it, at some point in the lecture I'm going to show you pictures of the ATP synthase. It is something that you perceive as one big protein, not as a complex that interacts with many proteins, but it is spliced together from different places. So yes, there are proteins that we perceive as single proteins and not as complexes that are coming from different genes. So very much this issue keeps in flux how many proteins we in human have. Some people, and now that has to do with alternative splicing, alternative construct formation. And again, the numbers are currently changing. So some people again say now there are 20,000 genes, but 100,000 proteins. So baseline here is not that well defined what a protein is. Typically it's a functional unit, or one way that we did in the last semester is a structured unit, so something that adopts a unique three-dimensional structure, and by that you can recognize it, and two proteins you can compare with each other. There are sequences that have unique features in terms of function and structure, here like peppermint and spearmint, and essentially the entire part of the lecture, the goal of the game really is to look at these two sequences and say there are differences that matter and differences that don't matter if it gets to the issue of peppermint versus spearmint, and then there are differences that have to do with the fact that this is in fact a cytochrome, that both of them are cytochrome P450s, happen to be very old proteins. Can I look at this sequence and see what is responsible for what? What are the binding sites for which reaction? Now the baseline message of the lecture is we cannot do that, but we're going to have to drill the problem down into certain aspects. We can predict aspects of function. And some of these aspects of function are really nowhere near that. One aspect of function has to do with how proteins interact, one where they are located. One is in fact something as simple as this. Is it important that this is PKP and this is PPS or something like that? So is that particular residue mutation relevant for function? Not necessarily for the peppermint versus spearmint function. When I say that change is relevant, it may be for the cytochrome P450 function. So all of these together give different answers that go toward the level of function. Now what is function for you? What is protein function? So we don't know what a protein is, but it's sort of something that is defined as having a common structure, having repeatedly the same structure, and having a common function. So what is function then? Let's look at an example. Alcohol dehydrogenase. Very important function for the people in this room in particular, because it helps you. I'm not talking about the beer intake from yesterday. I'm talking about the ethnic background. So I would assume that everybody in the group has DADH. In the previous lecture, that was absolutely not the case. So this is not the Asians don't have that. That is the reason why there is some issue with alcohol. ADH is a homodimer, so 94,5 residues. The homodimer is roughly sort of can see it here. It forms the same thing, comes together again as a dimer. This dimer formation is essential for its function. Its function is an alcohol dehydrogenase, so it does something to an alcohol. Now, in more detail, the function is defined here. There's an enzyme classification number. I'll get back into that in a minute. There's a size, and there is a description of what exactly the chemistry does. And there's another description here that has to do with the fact it is part of a pathway. And we get to that in a minute. EC number, the function of ADH. EC number is 1111. Oxyreductase is on the first level, the EC1. 1.1 is the CHOH group of donors. So we get into further detail here about how that particular chemical reaction is done. That's what this number is about. Next week, we're going to talk about, Tobias is going to talk about Go. And I'll get to that in a second. And the week after that, I'm going to talk about a method that Janet Thornton's group has proposed, which essentially tries to compute on enzymes by getting to this chemistry or describing enzymes through the chemistry. Now, this is a cake pathway.
Glycolysis. It's glycolysis, right?
That is written there, but how did you see that without knowing that it's written there?
I was looking at it yesterday, that's why. I had to do it yesterday for something else.
It also says on the
So that is clear to you that it's glycolysis?
Pardon?
Yeah, that's what I meant. But he obviously he he looked like he had not seen it. And I'm just impressed that you visually recognize there are about over 1,000 pathways, and you recognize this one.
Well, I did have to work with.
There must be a way for you to pull out some pathways that will confuse you. Yes, it was a remarkable hit. Funny how biology works, right? Anyway, indeed it's true. There are many, this is one particular one, and the fourth step is here, one path, one particular step in this path, and that gets into gene ontology. Gene ontology, in fact, unlike EC, tries to also capture the pathway. Gene ontology comes on different levels, but okay, let's get back to, before we get into gene ontology, another issue. So you could define function as something that happens on the level of chemistry, how the atom is bound. You could describe it as the biochemical, this is a transferase, or in this particular case before it's an oxyreductase. On the cellular level, it's a kinase. Or it is something that is part of the cell cycle, which again defines life. It's one of the processes that define life. It's developmental. It regulates at some point how this organism is going to look, whether the frog is going to have two heads or one. Whether it's related to disease or whether it's genetic and dominant. For the course of this lecture, we will define function, anything that happens to or through a protein. Is a definition again. Now, what really is function? On which of these levels you define function depends on your background. There is no answer. Function is not any of those. It depends on your focus. Our focus will be more on the chemistry of the issue. And our goal is to predict protein function from sequence. Now we didn't have that. So let's just say there. Function is known for, the statement that I make is function is known for 10 to 50% of all human proteins. Swallow that. Once you have swallowed it, my question to you becomes, why is the range so big? There's a factor of 5 right in there. And before I was sort of stingy on your factor of 5. And now I'm easy to give a factor of 5. So what's, yeah?
Maybe there are predictions for 50% of proteins, but it's only confirmed for 10.
How would you know what, so part of the important advance of computational biology is that it gets increasingly difficult to distinguish a prediction from an experimental annotation. We have more and more a lot of data that is very noisy. So we have annotations that give us something but not a clear evidence. And we can interpret those data only through the lens of prediction method or statistical or evidence integration methods, often Bayesian networks and the like. Essentially, that implies that you lose the clear distinction whether it's a prediction or not. You're right, that could be the answer. But in fact, in this particular case, it's not.
Then maybe it depends on how we count proteins. So if every isoform of a splice variant is...
That is, in fact, you're right, and we have another problem that comes on top of this categorization. Yes, you're right. Yeah?
Maybe protein has more functions and we know only one or two.
Very true. Another problem that is ignored. And it's a very big problem, because very, very few proteins are molecularly annotated with exactly the region that is relevant for a particular function. Most proteins have more than 2 domains, so most proteins will most likely have additional things. No, the major problem here is something on a different level. So previously I showed you the ADH example. So I annotate this protein to be an oxyreductase. And I have an experimental evidence that it's an oxyreductase. Now, do I annotate the function? Or do I actually, before I say I have annotated the function of that protein, really want the full EC classification? That already is one of the big reasons here for the spread. So we have, for many human enzymes, we have some idea that they are enzymes. Doesn't mean I know the function. For some fewer, but still a considerable number, we know that they are lyases versus oxide reductases. But for much, much fewer, we have explicit evidence experimentally how the biochemistry is done. And even for those for which we have the detailed evidence, let's look at titin, for instance. Titin is a molecule for which the longest protein in the human, over 30,000 residues, we know has many, 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 many domains, and for many of these domains we know what they do. But what the whole thing together does, it's not like an actin fiber. It can form fibers, it is part of that, but the entire molecular environment, the entire function of titin, prion, you talked about prions, is another example. We have a protein that is very well studied. We know so many things about the function of prions. We still don't quite know the molecular environment. We still don't quite know the molecular function of prions. Why are they there? Why are they positive? And that ultimately is the major part for this immense split here. But all of these things work together, you're right. So we way underestimate this. So I would argue that possibly both numbers here are overestimates because they have multi-domains and because some of these annotations in fact are predictions, not really experimental annotations. And this problem is getting increasingly difficult with the increasing databases. That is a problem. What we do also know, so let's get back to the people who have not been in last semester. Protein thrown into solvent essentially adopts a unique three-dimensional structure. The three-dimensional structure is important for its function. But we cannot do this in a computer. We cannot take a stretched-out sequence, do molecular dynamics, and predict the structure. We can do that for short fragments in some cases, but the most we can. The longest time frame is microseconds. Some proteins form on the level of minutes. We really do not know how far we are on that one. What molecular dynamics, however, can do is simulate, given a structure, mobility. And function is essentially something that has to do with mobility. Proteins function because they are mobile, because they move. Doesn't work very much on this one here. Sorry, but on this one you begin to anticipate, and that's one of the slides from Marco Punta. You see the movement, and the movement is functionally relevant. No protein is a brick, and the degree to which it's not a brick is exactly the way it functions. Still, we believe that order has to be there, and that function is possible only because you get the same thing again and again. The exception to that, you know, is disorder. So you have regions that define definition of an operational definition. We cannot do that experimentally, but an operational definition of disorder is you shine light upon this molecule twice, and you get two different solutions. Okay, red one here. Then very different than the blue one. In the blue one, you shine light at two different time points. You see the same thing. Now this one binds to the blue, the red binds to the blue, and upon binding it adopts a unique three-dimensional structure, sort of helix formation. And now you shine light twice to the blue, to the red, and you will see the same sort of helices. Induced fit. Disorder plays an important role in different ideas. Here's the induced fit idea. Here's the idea of bridging, of coming together. That is a coil here, with which you in fact by coming together can also join cells. What is not here is a domain linker, so you have a functional motif by creating flexibility. These arms can do different things. Another way in which you can imagine disorder to be important, and let's just look at this stretch out thing again. What could I do with an arm like this that I cannot do like this? What is the advantage?
Longer.
Yeah, and what is the advantage of a longer arm?
You can reach more far.
You can reach further. Yes, it's a great point. Unfortunately, I'm not aware of an example in the protein world where you need to reach further. Actually, it's true. So in this context, it is true. So in the context of the coil, where you have essentially two bodies of cells that approach each other, and the way they come to each other is by having coils wind around each other. By winding around each other to pull the cells. That has to be long. But this coil motif. For a completely disordered region, I'm not aware of an example like that. But what else could you do with reaching further? Yeah?
You could wrap around something.
That is the induced fit binding. That is sort of the idea you bind to something, right? Yeah?
Yeah, I wanted to say the same.
Oh, you want to say the same thing? There's something else you can do, right? I mean, come on, you know, imagine you take a subway, right? Just imagine a densely packed subway and you do this one. You create space, right? A space maker, filling material. The stuff that is where your presents you get from Amazon are wrapped in or something. There's these plastic buffer zones. And there are proteins that are disordered because of that. They obviously seem to be buffer zones. Another thing you can do is you can sense the environment. And now we're back to what you said in a slightly different context. So yes, you can reach further, not to actually grab something, but you can reach further because you can sense intrusion into your space. And that is another theme. So you wiggle around and sense some other molecule that comes. You're not actually acting to it, so you're not grabbing it. In that sense, you both of your ideas together somehow, but you sense and you send a signal, and that signal leads to some process. And that is also what we see. Disorder, in fact, is very important, and it's particularly important for eukaryotes. Every third protein in human has one long disordered region. Now, again, I said that protein function is intuitive, but it's not well defined. Metrics. What is a metric?
Some sort of measurement.
Metric is a measurement. That is true. Any refinement of a metric is a measurement. Yeah?
There's a formal definition and it has to comply with some characteristics, but yes, mostly it's a metric.
Can you name some of these characteristics?
And you can like calculate with it then, right?
That would be great, but a measurement is something you can calculate with, right? But yeah, what are...
To be comparable, so if you measure two different things, you have to be able to compare them.
Yes. So if A has a certain similarity to B, that is 50%, and then A is 50% similar to C, then you want to be able to say, or 30%, one is less than the other. So two measures, in principle, measure could be something else. And we will get to that. In principle, the word measure in itself does not necessarily imply that. It is suggestive, but it does not quite imply it. What is another? Does anybody know another characteristic of a metric?
The triangular inequality.
Exactly. This one here. So the fact that A is similar to, has a distance to B, and A to B and A to C implies something about the furthest distance that C and B can have. It has to do with distance. Now, let's look at typical measures that we have. For sequence, we can clearly measure E-values, pairwise sequence identity, and things like that. Is that a metric? Why not? Oh, come on.
Actually, for sequence identity is a metric, I think. We had proven it to be one.
That's an amazing achievement.
Maybe it wasn't exactly sequence identity.
But I'm off. Who has proven? I like this. I like this. I immediately write it down. I immediately, you know, whatever journal I'm the editor of, I'll publish it immediately. I'm looking for something like that. So we don't have a metric. And in fact, the major problem here is in a metric, the triangular equation, a metric needs distance. They don't do that. That two proteins are similar to each other, that the E-value is high, means that something. They are related. But that the E-value is low doesn't mean anything. Okay? So that is one of the major problems here. Now let's look at RMSD. Structure comparisons. Is that a metric? Why not? I'm just for the sake of it, I'm going to make you 100% wrong today. Why not? Anybody wants to answer the... He says not.
I also would say no, because there's no null level, there's no comparison to a something unrelated. It's always pairwise.
You don't have a distance, is what you're saying.
Yes. You have a distance from one protein to the next, but you need some sort of usual complete point of comparison.
And the two proteins have to same number of atoms to calculate the average.
Okay. So we have problems, but let's assume we compared all the proteins with a similar number of atoms, or not atoms, residues. Okay, let's just look at C alpha. So we look at all the proteins with a similar number of atoms. Then do I have a metric?
But it's a distance, right? So it should.
Is it? He says not. Or I'm not entirely sure. First you said not, and then you said yes.
I thought it was a pairwise distance between two proteins.
It is.
But pairwise identity is...
This is pairwise distance.
Take the mean over.
But it's still a distance. It's still a distance. So let's go to the question here. So we're talking about the same residues. The distance between these, or the similarity between these, is 3 angstrom. The similarity between those here is 2 angstroms. What does that tell me about C and B? We're talking about the same residues.
So it is like an NMR measurement, and we calculate RMSD between models.
Actually, it implies something.
Maybe that
it tells you that they're not identical, maybe.
So now we are back to Sabrina's point. So proteins have different lengths. But in principle, you could imagine that you can normalize by that. And there is a paper in Bioinformatics from Manfred Zippel that in fact introduces a metric for structural comparisons. It is possible. And the major problem really is the length. But you sort of can normalize them to the common length, and then you can come up with a measure that is indicative of it. And in his, he shows, so that is just the principle that he introduced in this bioinformatics paper, and in his methods he shows that he can actually use that to distance compare protein structures and come up with conclusions that are relevant for biology. What about function? Let's look at the first one here. One metric, or one candidate for a metric, is the enzyme nomenclature. EC, 6 numbers. The first digit, oxidoreductase, transferase, hydrolase, lyase, isomerases, ligases. And then we get into, here for lyases, the particular reaction and further detail and further detail. Yeah?
I'm sorry, but we couldn't even define protein function. Now I want to measure distances between functions?
No, be careful. So first I said that function is something that happens on all levels. Now I'm looking at one particular level. And this is a level somewhere between the chemistry and the biochemistry of the view. And this particular view, for that particular aspect, so I'm not looking at the cake pathway, I'm not looking at the role, but I'm looking at that particular characteristic of a protein. It's not, it is an enzyme, it's an ADH. That's not all it does, but it's one aspect of what it does. While I'm on this one aspect that it does, can I there define something that looks like a metric, is the question. So there is no principle no first. You can shoot me down, but not with a big gun. Yeah?
But it's like a tree, so you could define a distance.
On a tree, can you not?
Yeah, like go up 1, 2, 3, and then down again, or you just go up a little bit, and so they are more connected.
All it needs
distance, and then you can.
Yeah, exactly. So all it needs is that you have, if you have levels, it's a little bit more complicated. That much is clear. But still, all you need here is that the levels down here, the 4th digit, are comparable between. So whenever you get down to level 4, you have a sort of similar level of detail. And when you look at a tree, here in this case level 3, and then you know that proteins on 2 different parts of the tree of level 3, they are really distant from each other. And for the enzyme classification, that in principle is the case. Enzyme classification is such that proteins don't partake in two different parts of the tree. Sometimes they are wrong in this classification. So sometimes you find things that are down here and that essentially, in terms of role or in terms of chemistry, do something very similar. And that's what I'm going to talk about in 2 weeks from now. Yeah?
And also in cake pathways.
That is not.
Yes.
Totally independent of a cake pathway. So again, so the way I sort of stopped your gun is by saying, listen, what I'm talking about here is one particular aspect of function, that it's not related to the pathway, it's not related to where the protein does it, but how it does it, about the chemistry.
Reactions on the cake pathways labeled with those EC numbers.
Yes.
Then why does it happen that the reaction has 4 or 5 different EC numbers on it?
Oh, so why one reaction has that.
Then
you can have two
same enzyme with different classifications.
Yeah, that's what that is a limitation. Okay? The second part is a limitation. The first part is a lack of detail. Part of it can be that in fact two domains have two different enzymatic activities and have to come together, and they do that. Or that two proteins have to come together. So the function does not necessarily happen by two proteins binding, but by two complexes binding. And you immediately explain why you have many EC numbers. So there are essentially many molecular reactions, chemical reactions that happen simultaneously by bringing together different units. That would explain why many cake pathway dots, in fact, are not resolved further. The second one, why you have a jump between the tree, there's a limitation, okay? So how could we get over that limitation? One way of getting over that limitation, or one of generalizing, is attempted by the Go, gene ontology, and that in fact says, first of all, we have to classify everything. When we classify everything, we have different ways of classifying. One way of classifying is molecular function. That is like the EC. But it begins on a higher coarse grade level. And further down the tree of molecular function, at some point it will say enzyme or not enzyme. But above it, there are other levels. One level is as coarse-grained as cellular component localization, so where the protein is performing its function. And one is the biological process. Now we talk about cake. Okay, we have all of these here as an example on the biological process, DNA metabolism, on the molecular function. You go into a DNA-dependent blah, blah, blah. So there's helicase and things like that. And then we go into the particular nuclear component or nucleoplasm, so further detail, nucleolus, nucleoplasm. So detailed organization in the cell where the protein is performing its function. The important reality of Go is, and here are just a few examples for that, the important reality for Go is that now proteins are classified in two different places simultaneously. So the entire philosophy in Go is you can be at two different places in a tree. That's one statement. You're part of biological process, you're part of molecular and component. That's already the first split. But even inside of all of these assignments, you can be at different parts of the tree. Second statement, the EC classification, level 4 means something else for an oxyreductase and for a lyase. But still, it means the highest level of detail I can ever get at. The Go trees have very different depths. Some of them go to 8, some of them go to much shallower depth levels. And they are not intercomparable. That is a big constraint of Go. I'm going to rush through a few slides. Similar sequence usually implies similar structure. That was the content of the last lecture, and you can use that for comparative modeling. Then there is, in fact, the same sequence can have different functions in, for instance, different tissues. That's a different environment, essentially. And we have the example where you have completely different structures that have very, very similar binding sites, in this particular case here, and perform the similar enzymatic activity. There are chymotrypsin, cyptilin are examples for that. There are many examples. Proteases is just a family of things where you have an immense diversity underneath the same function that is maintained by a particular binding site. Some of the issues with the problem, one of the issues that you mentioned before, assigning domains, most assignments are done like this. So you know this is an ATP sulfurylase. You find another protein that has a similarity. You assume it is an ATP sulfurylase. You look up and you see that's a synthase, something totally different. You explain that by saying, oh, okay, the region where they're actually similar is not the region that has been annotated functionally. So the functional annotation, in fact, was this and that. And the red region that I used, in fact, to infer that function, why Go calls that this and that, is a mistake. And there are many spurious assignments like that. So in fact, this here in that view would be a hypothetical protein that does something totally different that we don't know. That possibly is part of what this does, and possibly not. So maybe an additional function in the context. Moonlighting, you know what moonlighting proteins are? Did I talk about that last semester? Yes, I did. Who in the room who was not in last semester's course knows what a moonlighting protein is? Yeah? Say it.
I don't know it.
Ah.
I thought that was the question.
Any idea? People not in the last semester, you know what moonlighting is? No. Working two jobs, essentially, to do two different things, and that already gives you the idea what it is. So proteins that in fact have two different roles. Sometimes these two different roles happen because you're in a different localization. Sometimes because you're expressed, and I showed you in a structural example for that, you're expressed in different cell types. Sometimes because you bind a cofactor. Or you have different oligomerization. So this is the ADH that forms a dimer, a homodimer, and this one is forming a tetramer, and it functions differently. Sometimes it is that you bind as a group versus you bind alone, and sometimes you in fact have many binding sites. Most proteins have more than two and more domains, so very likely that they have more than one binding site. Okay, let's get into the next lecture. Sorry for the introductory slides. Homology versus can you read that?
The syntax could use some work, but yes.
Well, come on. I have a protein alphabet. Oh. I'm very restricted here. It's amazing what our brain can do in terms of filling in white spaces. Why do we want to predict function?
Because if it works, it's easier than doing the experiment.
Yeah, much cheaper, and it's important. So one way of predicting function has to do with classification. We want to know that something is a kinase. We want to know that something is another unknown contributor to some pathway, fills in some roles. This is one view on all the protein kinases. And typically the kinases are classified by sequence similarity. And that brings us, some of you have seen that before. So very near each other of proteins, you can classify function according to sequence similarity. The further you go, the less that signal will be good, and you need machine learning to help you out. And this is particularly the case for function. So the idea of homology-based inference is clear. You have a protein for which you have experimentally annotated a feature. You have another protein for which you don't know the feature. They have a similar sequence. You say they have the same feature. That's homology-based inference. And what's the philosophy behind it? Why does it work?
Because in many cases, a similar sequence implies a similar structure, and the similar structure implies a similar function. That's
a very reasonable assumption. But it's also true, I showed you examples where you can have a very different structure and do the same function.
That's why I was careful with my wording.
We actually believe that the more important pull, or the more important reason, is not the similarity. What you say is true, but the more important connection is the evolutionary connection. And the evolutionary connection is one of history. And that ultimately... This is at 2:30, Tobias.
Okay, I don't know.
So, the evolutionary connection, let's just zip over it. So you're related by evolution, and that relation leads to a similarity in the function. Back to the article dihydrogenase, back to homology. So that means that through sequence comparisons, you in fact cannot infer exactly the evolutionary tree. We don't quite know the tree because we don't quite know the event, and that may lead us to unclarities how to infer function between orthologs, paralogs, and brings a lot of uncertainty into the equation. Now, there is the domain problem, we talked about that, there's the moonlighting problem, but let's just look at one particular aspect of function. So we're back to molecular function, we're back to EC classification, and let's ask how well can we infer function through homology inference. So we pretend that this protein up here we do not know, and we look up homologs here and we ask how often are we right. So in the first case, so we have some level of identity, we pick up things, and at some point we pick up things. Let's say our goal were to be to infer that this is a methyltransferase. So this is an enzyme, but it is a transferase, but we actually want to, we call functional assignment now, it is a methyltransferase. Okay? And here we would make a mistake. Here we would make more mistakes, and here we would get the answer. Now the big question is, where do we draw the line? When do we stop? So this, I can't even remember whether it's a made-up example or a real example, but obviously we can go further down, and the further we go down, the lower our accuracy becomes, but the higher our coverage, the more of the things that are also methyltransferases we get. Now the real question ultimately is, what's better, 66% accuracy at 50% coverage, or 38% accuracy at 100% coverage? Where to draw the line ultimately is the question. In this example here. How can we decide that?
You drew the line between two identical percentages, so it's not really good.
I did what?
You draw the line between two identical percentage identity.
That is interesting, yes. That's an interesting one, I have to change that. And so this suggests that it's a real example. So it's not a made-up example. That's an interesting point I've never seen. I don't know for how many years I've been showing this slide. I do know, yes I do know how many years I've been showing this slide. 11. And it never, anybody ever said anything. Good point. I have to change that, hopefully I remember it. Okay. But obviously, so one answer could be up here, so that there's nothing red appearing yet. But ultimately, again, how am I going to answer that question? Do you have any idea? So, you will encounter that in your research, in your later work. What are you going to do? One typical answer is, I take whatever is the first, because my experimental collaborators can only handle one answer. And don't make fun of it. It's a very rational approach. Is there any other way in which you could approach that by rationality? So you obviously need additional information or but you need to, most of the time, you will make an ad hoc decision. This is a lot what computational biology is about. And this is, in fact, Tobias will talk to some extent next week about how that can go wrong. So what Tobias will talk about is essentially the meta student. So what we did, we had a similar lecture a few years ago, and the task of the exercises was to develop a method that simply does homology inference of Go numbers. And it's very trivial, right? You have a data set of proteins for which you have the Go annotation. You run a sequence alignment against those. You pick out the one. You pick out the closest one or one of those, and then you say my protein has that Go number. And 3 different groups implemented that idea. The 3 different groups came together in December with their methods, and they described, just like you did before, they described what they had done in their exercises. And they all had exactly done what I told them. They were good students. They had listened to their supervisor. I find that very nice. And they all had exactly done the same. The only tiny dent in the picture, the only tiny problem in the picture was they did not get the same results. And it was not a matter, they did not realize it was partially a matter of a mistake in the programming in one case. But in fact, the difference between these methods, one was like random, and one was very good. The difference was that one got the details right, and the other one didn't. And all of these details were decisions of this type that I, as the supervisor, would have said, I don't believe it matters. Right? So decisions, unfortunately, we don't have good ways to make these decisions, but unfortunately, very often they matter a lot. So keep as long as you can in your head which decision was of this type that you don't have a rational behavior decision here, and possibly test alternatives. Usually you cannot do that for all your decisions. At least usually you can, for most you cannot. But keep it in your head and possibly deal with it later, or hope that you will be open enough to deal with it later. Let's now look at a specific challenge. The challenge of homology-based inference for subcellular localization. And that's work that Rajesh Nair, who is now at the FDA in Washington, started. So on the goal of predicting where a protein is in the nucleus, let's simplify this here. There's an extracellular space, there's a nucleus, there's a soup in between here, the cytoplasm, and there are mitochondria. And all the other details we're going to leave out. And we're going to essentially do the same thing that we did last semester. So we come from a comparison where we have 2 proteins that are 100% identical, and we're going to ask how similar are they in terms of their subcellular localization here. And we will somehow see that the further the sequence signal goes, the more we will find cases that don't have the same subcellular localization. That's the black line here, and the gray line indicates of all the things in the nucleus, the fraction that I find here is 100%. But when I find all nuclear proteins, actually I find all other proteins too, so at that level of classification, my accuracy is dismally low. And I will have some better payoff for that. For structure, those of you who had been here before saw the H-val, the HSP curve, where if you have 100 residues aligned, 33% of these, or 33 of those are identical, and these 2 proteins evolved naturally, then essentially they have a similar structure. Now, we, in fact, in order to test how this will work for subcellular localization, we have to take all the proteins of known subcellular localization, experimentally known subcellular localization, and sort of compare them to each other. That's the first part. How do I get all proteins of known subcellular localization? The slide says Swissprot. There's a Swissprot line that says subcellular localization. Is that what you take? Ever use Swissprot? Whenever you do that, so in many cases, before you do that, not look only at one example, look at many examples. So the point is, Swissprot also has a line that says probable or possible. So some of these annotations, in fact, are by homology. So if we took the ones that have been inferred by homology, and we want to use those to infer how well homology inference does, we would be very, very, very circular. We have to avoid that. We have to make sure that what we are putting in here is really experimental knowledge. Okay, now we have real experimental knowledge. We still have this issue of bias, right? We cannot use the entire set. We have to find some sequence-reduced subset. some set here shown in white that is such that no pair in that set is very, very clearly identical, to make sure that we're not oversampling some part of its space. And then we take this set and compare it against everything else. For a structure, this is sort of what we saw. The HSSP curve is in black here. Red are things that have different structure. Blue things that have similar structure. So red is all over, blue is all over, but at least the line is such, the density is not shown here. Let me show you the density. Okay, I don't show you the density. On a later slide. But the density is such that actually what looks highly red here above the curve is not that dense. Most points really are above here. So let's compare it to the subcellular localization story. What are the differences? And actually this one here is the new line. That structure, red different, blue same structure. That's subcellular localization. Blue, similar subcellular localization on the scale of 4, mitochondria, nucleus, extracellular, cytoplasmic, and red, different. Describe the differences.
The curve is exactly the same, right?
Yes.
The gray one.
That's the first statement. So the next statement from somebody else.
Well, for one, the points are not as segregated, or are more segregated there. On the right, there's more points to see. There, they're all.
Yes, that's a good point. So, again, in when you write a paper, introduction, methods, results, what you're doing now is you describe your results. You don't interpret and you don't put them into perspective. Some of the interpretation can go in, but only as long as it really has to do with understanding what you plot. And that is an important observation, that sort of information. And that is simply because you have more points. Okay? It's a relatively trivial point here. Yeah? But you cannot see that, so it's an important issue. Yeah?
And the red is more above. Also more above.
Yeah, that's difficult to see, but clearly you see it very much suggests that there is a lot of red here.
Yeah.
That's true. Anybody else sees any other observation? More extreme? No, anybody else? Part of anybody else is not the same.
That's a good advice too.
No, but yeah, it's true.
The points, blue points are also denser.
Yeah, but is there anything else that you see that is extremely different here on the right? So look at this.
Yeah, there seems to be a sort of pattern.
So we have blue dots here in cases, mind you, what we have here is a pair of proteins that have a different subcellular localization at 95% sequence identity over 400 residues. So we have very long proteins, longer than your average enzyme, most likely, so that could be a tin barrel, but it could be a two-domain protein. Both domains, almost entirely identical, 95% or more, still have different subcellular localization. This you absolutely do not see in that picture. These extreme cases, yes, so far we have looked at this higher density is true, but the very extreme cases here are really different. And you see that when you compare, no, that is not a comparison, but what I show you here is percentage sequence identity, accuracy versus coverage, log E value, and the HSSP distance. For all of those, you see that really it is very, very complicated to stay at 100%. One reason here is different tissues, why this could happen. There are other reasons, and that essentially have to do with the environment. Now, essentially the way to use those data, so what can you learn from it? When you look at that, or even more those are your results. So you asked, how well can you do homology-based inference according to sequence identity? You pick whatever you like in terms of measuring sequence similarity. Be it percentage sequence identity, be it blast eval or psi-blast e-values, and be it this HSP distance, which is essentially the distance from that gray curve I showed you before. Whatever your choice, how do you use that now? Well, you can publish it, but how can you use that for homology inference? What do you do with that when you publish a method? Is it too obvious and nobody says anything because it's too obvious, or it's too October, or? Can you, so when you have a method, the method has an input and it has an output. Does that define, let's first begin with the following, and that relates to the question that I asked before. Can you put a threshold? Can you say above that threshold, two proteins have a similar subcellular localization, below they don't? If so, whoever says you can from these plots? Nobody? I'm going to ask you why you cannot if you say, everybody else says I cannot, and it's the same question. You will not get out of.
But there was an example with 95% identity in different locations.
That's true. But still, we talk, so the great advantage of a computational biologist over an experimental biologist is we can be right 95% of the time, and we still get, and when we do that for millions of points, we are right millions of times, or thousands, many thousands of times, and we're still not bad off. We're still great with all these mistakes. If you remember, biologists, they make one go and they make a mistake and bye-bye birdie, right? So we can make many, many mistakes. So it's a probability thing.
Okay.
Yeah?
So you probably can, but not with.
Where would you put it?
The cutoff, you mean?
So that's the question. Would you put a cutoff?
Yeah, it's on the application.
So you want to essentially infer subcellular localization by homology. For instance, one application could be, I told you that Swissprot has a field that says by homology. You could simply test how well that does, right? Because now, in fact, you reassessed it. They haven't done that. You're the first to, this was really the first time that was done. Nobody before had ever used it. Before, what people had used is 30% sequence identity is a high number. Everything above has a similar one. Everything below, or psi-blast e-value 10 minus 3, that's some point here, which you see is absolutely wrong. But you could, for instance, use that to assess the database, to clean up the database. How would you do that?
Well, you go through the database and for every protein, calculate whatever your preferred method of calculating it is, and then compare it to the threshold you set, and then say yes or no.
That's my question. Would you use any of these curves, whichever you choose? Would you use those to define a threshold? And if yes, if your answer to that question is yes, then tell me where the threshold is. If your answer is no, then tell me what else you would use them for. If your answer is you cannot use those curves, guess you can use them. I don't agree with you. You have to try again to think. So, threshold or not, who has an answer to that? Whatever your answer, it's fine, just defend it. It's a valid answer. Okay. Let's do that differently. We have two halves of the room. They're not equally split, that's true, but they can talk to each other without moving the camera. So you guys discuss threshold. Which threshold, why threshold? You guys discuss on that side of the room. You discuss why not choosing a threshold. I give you 3 minutes, and then you present your solution.
So we choose not to put a cutoff.
Yes, and you don't have to argue why you don't choose. By default, you don't choose a threshold, but you argue what else you use.
Okay.
How you use these curves. Cutoff.
So, well, for myself, I'm thinking it doesn't really matter Because
yes, so the group agreed on that because he pushed them. And then I said that is not acceptable. So now he changed the group version to my own.
I mean,
whoever thinks differently before you defend it. The others were convinced by you anyway. So if you put a threshold, let's just take this one here, because for the sake of the argument, let's just put the point.
If I had to choose anything, I'd choose H as a speed distance, and I'd put the threshold on like 95% accuracy and 20%.
But this is not a random point. Now you argue for a particular, you turn around full circle. You're too flexible. Let me make the point here. So very often people have the tendency to say two lines intersect. That's a well-defined point. That statement is true. I would define this as a threshold. The problem with that threshold is you clearly see is you put the point in a position where a tiny mistake. So you're in the steepest line of the curve. And the steepest line of the curve is defined as you make a tiny mistake, or you move a little bit here, a little bit here. Makes a big effect. And you typically don't want a threshold to be such that if a little bit of a different measurement gives you a totally different answer, you don't capture that. So putting the curve, the threshold somewhere here, where the line is flat, is much more rational. Now, I'm fine, I'm not entirely sure. I believe we put it there at some point that here, because we argued that there was another dip here, so that for us was the, we looked at, we simulated the background, we believed this was not an error, there was a real dip here, so that's why we put it somewhere here. But the important point, take-home message here is never put it into extreme points of your curve. And again, choosing thresholds will be something that we all will do again and again. And that, by the way, is another reason why this is not necessarily the best thing, because it's extremely steep. If you chose an E-value in PsyBlast, then now the reason that speaks for the H-val as opposed to this is, say you choose the 90% point here. At that 90% point, you get something like 50% coverage. Here at the 90% point, you get higher coverage, I believe.
Yeah.
Somehow.
2%.
It's small. So that is wrong actually. So that would be speaking for PsyBlast because it's easier. You guys, no threshold. How do you use the data? Oh no, really? Come on, but you had it on your.
We just take the best. Like we just take the.
We take the threshold, but you're not allowed to take a threshold. How can you say we take the threshold?
The best.
But that's the threshold. Oh, the best hit.
The best hit.
Ah.
But then, if you don't really have a hit, a really bad one, you're wrong.
The best hit is so the best hit. So exactly. You already have my argument against it. But you can simply cast. Again, you sort of can pseudo, but you can cast that into a probability. Right? Accuracy is relatively, maybe you have to do some normalization here, but essentially, and you know, the normalization could also define is in some sense a threshold, because the lower end should be 0, not 50%. But in principle, you can take the curve as it is, whichever it is, and make some probability function out of it.
Yeah, but that's like, I think that's like chickening out, because then you say, yeah, then I have a probability.
Wait a minute.
The user has to say, oh, now I have to define a cutoff. You know, at some point, you have to decide whether the 50% then is true or false. You know, then you have to define a cutoff.
I take that point, but your task was not. But anyway, I take that point. But where I don't take that point is if you combine these two approaches, and that's what you do in reality, right? You make something like a threshold. But if you combine these two approaches, you're actually more flexible in terms of moving the threshold. And you say, okay, I decide that the answer is clear. and within that framework of the answer is clear, I have a difference between here and here. And that's an important reality you want to capture in your method. You don't only want to have gray and white or black and white. You actually also want to say, this I am much more sure. It's still not 100%. I have cases where it could not be, but very few. And that has to be expressed by the method. That's a reality that comes out directly of these graphs that you have to put into your method, give to the biologist, right? Yeah?
But isn't this what I do when I say my method is like 95% accurate?
Oh, wait, sorry. Yeah.
Isn't that what I essentially
Yes.
said?
But that's what I asked you to
Do
do.
I have to spell it out too?
That's what I asked you to do. Actually, that is not what most methods do. Most methods give you yes or no. Experimental methods typically don't have the idea of probability, and even computational methods typically give you one state. And even in fact, lots of them, no matter how often I run around and preach this in my lectures, even many of our own methods still ignore this reality. Because ultimately it has to do with the complexity. In some sense it brings it back to what you said. I have to make a decision. But still, there's an important piece of information in here. And you can even tell the user, above a certain probability threshold, trust it. Okay, let's move on. Now, the problem is that we in fact have something here that is specific to a problem. We had this issue of structure. What is homology inference for structure? Now we have the issue for homology inference for subcellular localization. Will it be the same threshold for enzymatic activity? So I established when I can homology infer that two proteins, threshold, probability, mixture of things, that two proteins have a similar subcellular localization. Will that be the one that I want to use for enzymes, or will I use the one for structure? Or will I have to do a new one?
Can we return to this probability distribution as well? For example...
But how do I get it?
Okay, I don't know. But I mean, for something as generic as a database, I mean, I don't know what the research is used for.
But my yes. My only question to you now, when I give you the task of doing your master thesis, you have 6 months. Now you could build a database right ahead using the subcellular localization or structural similarity, probability, and put it into a database, or you could redo what I just showed you for subcellular localization. What would you do? What do you believe you have to do? Unfortunate reality is you have to do it again, and that's the frustrating reality of function. That essentially every aspect of function you have to redo and redo and redo. So what you guys chickened out with by taking the first is something that Tobias will also talk about, which has to do with you have to make a choice at the end of the day and you cannot do all of them. So you take the first, no matter what. And since the first more often than not is something that is helpful, I can live with that choice. That's reality. But in detail, for the background of the database, for assessing how similar the assignments are, how good the assignments are, you need to go through the whole procedure again. And the whole procedure, again, we talked about, I brought you the enzymatic classification. And here, by the way, is another example for having a very different structure that does the same enzymatic activity. Conservation of function. So here's a slide that was published by people, and they, I should have removed the authors. Identity classes means structure comparison, and that is percentage sequence identity, and blue means essentially that they have the same structure. Here, blue means that they have the same keyword, functional classification from Swissprot. Here it means they have a similar binding site, and here it means that they have a similar enzymatic activity. In fact, the blue is all 4 enzyme digits, the red is the first 3, the green, the gray is the first 2, the green is essentially the first digit. So, The numbers read 95, 85, blah, blah, blah. One clear conclusion is that enzymatic activity is fairly well conserved. Yes?
I'm sorry, what are the digits again?
The classification of the EC number. So the first digit is oxidoreductase, transferase, blah, 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 ligase. The second digit is the ligase is the last one. The second digit is the molecule and blend. Okay? So the green means that, in fact, what is conserved is they are both transferases. The gray means they are methyltransferases. That's the previous example I had. And the blue means they are really EC number 2113. The S-methyltransferases, the particular S-methyltransferases. So that's a paper that was published. Wait a minute, where do I have that? Okay, whatever. No, I'm going to, Here we are. So that suggests that enzymatic activities were conserved, and I felt that I didn't believe that, and I believed that in fact the problem was what they did is they took all enzymes of non-function and did an all against all. And my argument was, well, maybe you're right, maybe not, but you cannot do that. Because by doing an all against all, you essentially square the bias. In terms of percentage of families in the bias, if you do all against all, you see something like this. If you do an all against all in an unbiased way, so you do a subset, unbiased subset, and then do against all, and you look at family sizes, then you see this. What does that suggest to you? So we're looking at the feature family size, and we have one distribution blue and one distribution red. Which one do you believe, even if I had not labeled them? Which one? Again, I'm looking at the distribution of families with 1, 2, 3, 4, 5 members. Which one do you believe is the more likely real answer? Red or blue? No, not cheating. The way you smile, I know you're cheating.
Doesn't it make sense that a family with a lot of members is more distributed because it has a lot of members, and each of the...
this one says, the blue says that there are many, 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 many small families and few very large families. The red one says that the number of families or the number of members in a family is completely spread out. You have the probability of having a small family is as high as the probability of having a big family. Which one is more likely?
So there's not the percentage of proteins in the family, but just the family?
That's the number of proteins in the family. That's the distribution. In this particular case, the percentage of all, but yeah?
So I would think that the blue one is right.
Why?
Because it makes sense that there are some families that have lots of similar proteins and some which are...
For who? It makes sense. What does it, can you define what you mean by the word it makes sense?
I don't know, it just...
feels good.
Yeah, right.
Anybody else who has a more rational answer than feels good? I'm not... it's good that you say that. It's just, I want more activity, yeah?
Maybe it just doesn't make, for most proteins, it doesn't make sense to have a big family for it, because
yeah, but that's the same.
Have like 5.
It doesn't feel like it. I mean, you
have like 5 proteins and they do this function. Why do you need 50 more doing this?
Well, some families do.
Yeah, some, but not a lot. For most proteins.
But that's exactly what he said. I mean, I'm not disagreeing with you. Yeah. I'm only not hearing anything new.
What about this scale-free thing?
What is that?
Happens everywhere in biology.
Yes. Whenever you have growth under competitive conditions, you have the Zipf law. That's exactly the Zipf law here, or something that looks like a Zipf law. So whenever in a computer you simulate growth of a city, you simulate growth of families, the blue is the sort of thing you get without any further assumption. And this is just a competitive phenomenon. It's not because it makes sense. It makes a lot of sense for you, because this is what we observe all the time. And we observe it because that is sort of the natural state of the affair. But you have to imagine, remember, this is a competitive environment. In a non-competitive environment, if we would not, as living beings, compete for resources, we would actually see the red. Or the probability doesn't make more or less sense. That's why I attacked you on that. But it's what we, in fact, your word of it makes sense is a map of that's what we observe all the time. And that is the zip law. So you immediately see, if you don't know anything about proteins, you immediately see there's something fishy going on with this view, the biased view. That cannot be true. So now, okay. I want you to remember every single point on every single curve. For the ones who are new, for the ones who are old, well, you know it. But let's go through it one at a time. It's a very, very complicated slide. You don't need to remember it, but the basic message that I hopefully get across, I'd like you to remember. This one here is percentage sequence identity. This is percentage of protein pairs. Dark blue is that is darn. So yeah, now it's impossible for you to take them apart.
This is the upper one.
So this means that the first EC number is conserved. The light blue means all 4 EC numbers are conserved. So the entire detail of the enzymatic activity. These set of light and dark blue are for the unbiased view. This is for the biased view. Now we get to the point that I showed you in the slide before. So that it is an enzyme, a transferase, in fact is largely conserved even down to levels of 20% sequence identity. If I use the unbiased view, you immediately see that at 20% sequence identity, essentially you have a random signal. While if you do want to have conservation, you have to cut off somewhere up here. So the biased versus the unbiased. The tiny little difference of, rather than doing database against itself, use an unbiased part of the database and compare that against everything, gives a hugely different view. And as I believe I have convinced you with a slide before, this is the likely view. It's the more likely to be right view. And it makes a big difference. And again, so here in this particular case, there is a point to be made about E-value, psi-blast, but ultimately, here, that was the point that I do want to dwell on in a moment. Conservation, those are the numbers, sorry. The conservation of structure in terms of an HSP distance, let's take this one here, is in green. So the HSP distance is defined by essentially nothing below, nothing above is minus, so that's why this is very close to 100 here on the right hand side of this curve. For subcellular localization you get the blue, so that is further away. You have non-100%, you make mistakes at much, much higher levels of sequence identity. But you still can define a threshold, you can still define a probability. For enzymatic activity, here EC1 in orange, and EC, all 4 EC numbers in red, it deteriorates earlier and further. The news here is, if you want to build your database about enzymatic activity, if you want to check how good the classification Swissport are, you really need to redo these curves. And that is a bad news, but now we have it for EC number. Now the next problem will be to do it for Go, because Go we cannot compute. For EC numbers, we can at least have a clear comparison of what 4 or 1 means. For Go, we no longer have this option. So that's one dead-end road. Another dead-end road that we hit is the story here of ER and endoplasmic reticulum and Golgi, where Kaas showed that even for those 2 different compartments, which essentially is subcellular localization, and another example for cell cycle, we have to redo curves, and then ultimately the goal of that was to in fact use these curves to build a database that then was used, this one particular cell cycle DB, but we need more details in order to build these databases. And every single time we had to sort of quickly redo the curves, and then we could use that to build a database. Now, homology-based inference, how much now can we do for human? And that is back to the 10 to 50% number. Where do these numbers actually come from? And one way of doing that is you now assume that everything is an enzyme, so all the functions are enzymes, and you assume that a certain level of mistake is allowed. So for the enzymatic activity, I know the level of sequence identity at which I get 90% accuracy. And I say 10% error is fine. Now my question is, how much of human can I do at that level of 10% error? The answer to that question is, so 10% error relates to this point here. No, wait a minute. Yes, 10% error relates to this, and for that point, I sort of get to a point of 20%. So that is if every annotation in SwissProt is right, 20% of the human proteins have an annotation at that level of error. If I accepted 90% error, so 10% accuracy, then my point would be 50%. Okay? That's on the level of getting all 4 digits of the EC right. So you made that point. This ignores the fact that proteins have several domains and that the annotation may be on a different domain. This ignores the fact that not everything in SwissProt really is an annotational function. It is just one particular record of SwissProt. So clearly it's an overstatement, but this gives you an idea where the number comes from. And I believe with that I leave it for today. Next week Tobias is going to talk about the meta student, so how we can use a simple homology-based inference of Go annotations and the kind of improvement over the simplest way with machine learning. And then next week, the week after that, I'm going to talk about this approach of computing function through chemistry and how we can infer function through motifs. Thank you for your attention.